Welcome to foodfeedinfo.com. This is a part 2 of carbohydrate metabolism lecture. In this lecture, we will cover KREBS cycle, which is also known as tricarboxylic acid cycle or TCA cycle. This cycle is also known as citric acid cycle. At the end of glycolysis, we have pyruvate as an end product. This pyruvate is converted into acetyl-CoA, and that acetyl-CoA enter into TCA cycle. The conversion of pyruvate into acetyl-CoA require pyruvate dehydrogenase complex enzyme. The combined dehydrogenation and decarboxylation of pyruvate to the acetyl group of acetyl-CoA, it requires 3 enzymes and 5 coenzymes along with 4 vitamins. The pyruvate dehydrogenase complex is a combination of 3 types of enzymes: E1, E2 and E3. The E1 is known as pyruvate dehydrogenase, whereas E2 is dihydrolipoyl transacetylase, and E3 is dihydrolipoyl dehydrogenase. The 5 coenzymes are thiamine pyrophosphate, NAD, FAD, lipoate and CoA, whereas the 4 vitamins are thiamine in the form of thiamine pyrophosphate, niacin in the form of NAD, riboflavin as FAD and pantothenate in coenzyme A. So this enzyme cause this dehydrogenation as well as decarboxylation and convert this pyruvate into acetyl-CoA. Pyruvate dehydrogenase complex, as I have told, is composed of multiple copies of 3 enzymes, the E1, E2 and E3. E1 catalyzes first the decarboxylation of pyruvate and producing hydroxyethyl thiamine pyrophosphate, and then the oxidation of this hydroxyethyl group to an acetyl group. The electron from this oxidation reduces the disulfide of lipoate bond to E2, and the acetyl group is transferred into thioester linkage with one SH group of reduced lipoate. E2 catalyzes the transfer of the acetyl group to coenzyme A, forming acetyl-CoA, whereas E3 catalyzes the regeneration of the disulfide or oxidized form of lipoate. Electron pass first to FAD, then to NAD. The NAD produced during this reaction, it transfer its electron to oxygen that ultimately generates 2.5 molecules of ATP per pair of electrons. So as a result of this, we have acetyl-CoA that enter into TCA cycle. So what is TCA? Before going to TCA cycle, just give you an idea about respiration, because burning of glucose in the body is similar to that of respiration. So as a result of respiration, there is a production of carbon dioxide and water. Similarly, in the body, when glucose is get oxidized, and that is occurring within the cells consume oxygen and produce carbon dioxide. This process is termed as cellular respiration. So during the cellular respiration, there are some stages. During the first stage, the organic fuel molecule like glucose, fatty acids and some amino acids, they get oxidized and they yield 2 carbon fragments in the form of acetyl group of acetyl-CoA. In the second stage, the acetyl groups are fed into the citric acid cycle that causes oxidation and results in the production of carbon dioxide. The energy released as a result of this oxidation is conserved in the reduced electron carriers NADH and FADH2. While in the third stage of respiration, these reduced coenzymes are themselves oxidized, giving up the protons and electrons. The electrons are transferred to the oxygen, which is the final electron acceptor, so via a chain of electron carrying molecules. So this is known as respiratory chain. So in the course of electron transfer, a large amount of energy released is conserved in the form of ATP by a process called oxidative phosphorylation. So in this lecture now, our major emphasis will be on TCA or tricarboxylic acid cycle or citric acid cycle. So citric acid cycle is a common pathway for the oxidation of carbohydrate, fat and proteins. It provides abundant ATP for body and also provides substrate for respiratory chain. TCA cycle is amphibolic in nature. It shows both catabolic and anabolic activities. If we talk about the catabolic activities, that is basically due to the final or common oxidation of a different type of feedstuffs, different types of nutrients in this cycle. So that's why it is called a catabolic. Whereas this TCA cycle, it provides substrate for the synthesis of non-essential amino acids, glucose, fat, heme. So due to this reason, it is also known as catabolic in nature. So here a little more explanation about this anabolic role. If we talk about this citrate, citrate is used for the synthesis of fatty acids. Likewise, in the cycle, alpha ketoglutarate is used to synthesize glutamate and oxaloacetate that give rise to aspartate. So thus non-essential amino acids, they can be synthesized through this cycle. Likewise, heme synthesis occur through succinyl-CoA, and glucose synthesis from this cycle occur as a gluconeogenesis process when malate is converted into pyruvate and it combined with the through this oxaloacetate. If we talk about this cycle features, so pyruvate after conversion into acetyl-CoA enter into TCA cycle. This cycle is strictly aerobic in nature, and that cycle is going on in the mitochondria. During the TCA cycle, the acetyl-CoA get oxidized into carbon dioxide and water, and there is a production of energy in the form of NADH2, the hydrogen equivalents, and it provides free energy to the tissues. Oxidation of NADH2 and FADH2 in the respiratory chain leads to the generation of ATP via oxidative phosphorylation. This is a complete cycle picture. Acetyl-CoA enter into the cycle and it is converted into citrate after combining with the oxaloacetate. Then this citrate is converted into cis-aconitate, that converted into isocitrate. Isocitrate is then converted into oxalosuccinate and then alpha ketoglutarate. Alpha ketoglutarate is converted into succinyl-CoA and succinyl-CoA into succinate, then succinate into fumarate and malate, and finally into oxaloacetate. This oxaloacetate again combined with the acetyl-CoA and enter into the citric acid cycle or after conversion into citrate. Here a little detailed description of citric acid cycle through chemical formulas. Just let you know about the importance of the citrate, because citric acid production in this cycle is of industrial importance. This citrate is used in a number of soft drinks, and also it provide a fruity flavor or tart when used in different type of drinks. It is also used as a plasticizer and foam inhibitor in the manufacture of certain resins, as a mordant to brighten colors, and as an antioxidant to preserve the flavors of food. So citric acid production in the industry through this cycle have a lot of applications, and industry is mainly using a fungus namely Aspergillus niger to produce this citric acid. The substrate used for this fermentation process is usually inexpensive sugars. They are used like this beet or molasses. They are used to produce citric acid. Now come to the point of this citric acid cycle again. Look at the diagram. The citric acid cycle is composed of 8 steps. The first step, if we start from the acetyl-CoA, that is converted into citrate when oxaloacetate is attached with the acetyl-CoA. So this is basically a condensation reaction, and it requires the enzyme citrate synthase. This is an irreversible reaction, whereas the conversion of then citric acid to cis-aconitase and then into isocitrate, they are reversible reactions. Then from isocitrate to alpha ketoglutarate conversion is again irreversible reaction, and then alpha ketoglutarate conversion into succinyl-CoA is again irreversible reaction. The conversion of succinyl-CoA into succinate, that is the 5th step, it is reversible, like the conversion of succinate into fumarate, and then fumarate into malate, and malate into oxaloacetate. All these are the reversible reactions. So out of these 8 steps of TCA cycle, only 3 steps they are irreversible. The first, oxaloacetate and acetyl-CoA conversion into citrate. Second, isocitrate conversion into alpha ketoglutarate, and then alpha ketoglutarate into succinyl-CoA. Now we will discuss each step one by one in little detail. The first step is condensation, where acetyl-CoA when combined with oxaloacetate result in the production of citrate. So this is the first step, and product is citrate, enzyme is citrate synthase. In the next step, the citrate is converted into cis-aconitase, and the enzyme required during this reaction is aconitase. So this cis-aconitate is an intermediate, and that is converted into isocitrate. So this is the completion of the second step. Citrate is converted into isocitrate. So in the first part of this second reaction, there is a dehydration and formation of cis-aconitate, and in the next part of this reaction there is hydration and finally the production of isocitrate. So both are reversible reactions and they require enzyme aconitase. In the next step, step number 3, isocitrate is converted into alpha ketoglutarate. The enzyme required at this stage is known as isocitrate dehydrogenase, and this step is oxidative decarboxylation step. There is a removal of carbon dioxide at this stage. Then this alpha ketoglutarate is converted into succinyl-CoA. The enzyme required for this reaction is alpha ketoglutarate dehydrogenase complex. At the same time, there is a requirement of almost the same type of coenzymes and enzymes which we have discussed when pyruvate is converted into pyruvate dehydrogenase pyruvate is converted into acetyl-CoA with the utilization of pyruvate dehydrogenase complex. So at this step there is also a decarboxylation, and finally there is a production of succinyl-CoA. So this is the completion of the fourth step with the formation of succinyl-CoA. Succinyl-CoA then converted into succinate. At this step there is a formation of one ATP in the shape of GTP, guanidine triphosphate, and this is a substrate level phosphorylation, and enzyme required at this step is known as succinate thiokinase, or we can also say as a succinyl-CoA synthetase. So that result in the production of succinate. Then in the next step this succinate is converted into fumaric acid or fumarate, and this step require enzyme succinate dehydrogenase. This step is a dehydrogenation step that result in the production of FADH2. So this is the completion of the step 6. The step 7 is a hydration step where fumarate enzyme is required, fumarase enzyme is required that causes the conversion of fumaric acid into malic acid or fumarate into malate. During this reaction there is only hydration, and in the next or the last step of this tricarboxylic acid cycle there is a dehydrogenation and malate is converted into oxaloacetate, and this step require malate dehydrogenase. As a result of this dehydrogenation there is a production of NADH2 and the product is oxaloacetate. So this oxaloacetate continue the cycle, or it can also go to the cytosol during the requirement of the glucose in a step of gluconeogenesis, or it can combine with acetyl-CoA again to continue the cycle. So these 8 steps complete the TCA or citric acid cycle. Next comes the energetics of TCA cycle. There are 5 main steps that result in the production of ATP or energy. The first step is isocitrate dehydrogenase that actually is converting this isocitrate into alpha ketoglutarate, and there is a production of NADH through this respiratory chain, and the total ATP produced they are 2.5. In the next step is that is catalyzed by alpha ketoglutarate dehydrogenase complex. So here again there is the oxidation of NADH by respiratory chain and production of 2.5 ATPs. The third step is catalyzed by succinate thiokinase that basically convert this succinyl-CoA into succinate, and there is a substrate level phosphorylation and production of 1 ATP in the form of GTP. The next step is succinate dehydrogenase is catalyzing the reaction, and succinic acid is converted into fumarate, and again there is a production of 1 FADH2 that get oxidized by the respiratory chain, and there is a production of 1.5 ATPs. The last or the fifth step is the malate dehydrogenase is the enzyme that catalyzes the reaction and then convert malate into oxaloacetate. Again, the production of 1 NADH, and this get oxidized through respiratory chain, and there is a production of total 2.5 ATPs. So in total, if we sum up all these ATPs, there is a production of 10 ATPs. So these 10 ATPs are from one acetyl CoA molecule that enter into the citric acid cycle. So if we talk about the glucose and its conversion into pyruvate and acetyl CoA, so from an hexose sugar there is a production of 2 acetyl CoA, so that enter into the TCA cycle. So we can say there is a net production of 20 ATPs through this citric acid cycles. So now we will conclude with the energetics of glycolysis and TCA cycle. If you just remember the previous lecture, we have calculated the energetics of the glycolysis. So in glycolysis there are the 3 steps where there is a production of energy in the form of ATP and NADH. The first is at glyceraldehyde 3-phosphate dehydrogenase, there is a production of NADH, and depending upon the utilization of this NADH through different shuttles, there is a production of either 3 or 5 ATPs. If NADH get oxidized through respiratory chain, then there is a production of 2 NADH, and in total we say 2.5 ATPs. And if NADH is utilized through glycerophosphate shuttle, then there is production of only 1.5 ATPs. So in total 3 ATPs, because 2 NADH are produced at this step. Similarly, at phosphoglycerate kinase and pyruvate kinase step, there is a substrate level production. There is a production of 1 ATPs, and that get oxidized at substrate level, and we get 2 ATPs, and due to 2-triosis there is a production of 2 ATPs. So in total we say there is a production of 7 or 9 ATPs during the aerobic oxidation of glucose. However, under the anaerobic condition there is only production of 2 ATPs. There is also a consumption of 2 ATPs during the first 2 steps. The one is at hexokinase and the other one is at phosphofructokinase. So in total, or the net amount of ATPs produced, they are 7 minus 2 is equal to 5, or 9 minus 2 is equal to 7. The values within the parenthesis indicate actually the total the values within the parenthesis actually indicates the old version of ATP's production. We usually suppose that 3 ATPs are produced through NADH and 1 from simple ATP. So that's why I have given the values in the parenthesis, keeping in view the old practice of considering the production of 3 ATPs from NADH. Energetics of TCA or tricarboxylic acid cycle. So there is a production of 20 ATPs as we have discussed in the earlier slide, but in total if we count these ATPs production from pyruvate dehydrogenase step, that is conversion of pyruvic acid into acetyl CoA, so there is a production of 2 NADH. Again, if it get oxidized through this aspartate shuttle, malate aspartate shuttle, there is a production of 2.5 ATPs per NADH. So in total there is a production of 5 ATPs. Then there are the 3 steps within the citric acid cycle where there is a production of NADH. So if we calculate, in total there is a production of 6 NADH, and if each NADH get oxidized through this malate aspartate shuttle, so it will give 2.5 ATPs per NADH. So the total NADH will yield 15 ATPs. Then succinate thiokinase is the next step. There is a substrate level oxidation, and there is a production of 1 ATP because there are the 2 acetyl CoA in the cycle, so 2 ATPs production. and the final or the next one is the succinate dehydrogenase, again oxidative phosphorylation through this malate aspartate shuttle, and there is a production of FADH2. So due to 2 acetyl CoA there are 2 FADH, and each FADH2 will yield 1.5 ATP when it get oxidized through malate aspartate shuttle, and we will have in total 3 ATPs. If we sum up all these ATPs, the value become 5 in the first step, and then 20 through citric acid, and in total we have 25. The values in the parenthesis, they are actually the old values, where we consider 3 ATPs through NADH and 2 ATPs from FADH2. So it's the summary of this energetic efficiencies or energy production when one glucose molecule is completely oxidized into carbon dioxide and water. So there is a production of 30 ATPs, 25 during the citric acid cycle, and 5 or 7 during the glycolysis. So 25 plus 5 is equal to 30, or if we consider 5 ATPs in the glycolysis, then we have a value of 32, whereas the old values, they are 38. But we will consider only the production of 30 or 32 ATPs as a result of oxidation of per mole of glucose under aerobic conditions. Whereas under the anaerobic condition, when glucose is converted into lactate as a result of anaerobic condition, there is a production of only 2 ATPs, because the pyruvate will not be converted into acetyl CoA, rather it will follow its conversion into lactate. So there will be production of only 2 ATPs. So as a summary, 30 ATPs will be produced during the complete oxidation of glucose under aerobic condition, and only 2 ATPs under anaerobic conditions. Little bit about regulation of citric acid cycle. The overall control is by the rate of conversion of pyruvic acid to acetyl CoA, and number 2 is by the flux through citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase. These are the 3 main enzymes that regulate this process. So these flux are mainly determined by the concentration of substrates and product. So either substrate is there or product is there, that will determine the fluxes. The end product like ATP and NADH, they are inhibitory. If they are present, they will inhibit, while the substrate, if NAD and ADP are there, so they will stimulate the continuation of the cycle. And the production of acetyl CoA for the citric acid cycle by pyruvate dehydrogenase complex is inhibited allosterically by the metabolites, so that signal a sufficiency of metabolic energy, because when there is an excess or sufficient amount of ATP, acetyl CoA, NADH and fatty acids, so they inhibit this PDA, and they are stimulated by the metabolites that indicate a reduced energy supply, like if there is a presence of AMP, NAD and CoA, so they indicates a reduced energy supply, and they will stimulate the production of acetyl CoA for the citric acid cycle by pyruvate dehydrogenase complex. So this is all about this citric acid cycle, the energy production during the citric acid cycle. So in the upcoming lecture we will discuss about pentose phosphate pathway of glucose oxidation. Thank you for watching, and stay tuned with www.foodfeedinfo.com. Thank you.